Alright, hello and welcome back to Jono's lab. This video is going to be maybe more boring and less produced than most, not that that's at all a high bar, but there's been a fair few things that I've been messing around with since my previous video that are all going to take either a long time to come to fruition or are too small to justify even creating a short video on. So I thought instead what I'll do is I'll go through my photo gallery and try and catch you up on a couple of the other things that have been going on. So I think the most previous video is measuring caffeine content at home. So there's a few others on my blog, but there's also lots of things that won't even make it into that. So I'll just start from that caffeine and skim past things that have been going on. Okay, so this is, I believe it's ammonium nitrate, but messing with exactly what concentrations and how fast or slowly you cool it. You can get some cool colors in polarized light microscopy. So that's with two polarizers, two linear polarizers at right angles above and below the sample. So that's kind of fun. Let's see. Okay, that's trying to transform my attempt at a green plasmid into BL21 instead of the original, what is it, MM294 strain that I had. Long story short, that also doesn't really show much expression, if any. It's a bit of a pain. Okay, accidentally found out that messing with the microscope filters and things, I could get some pretty cool like dark field style pictures of some of these crystals. That's pretty fun. Okay, zooming. Here's the artwork with the Pseudomonas. So the red and the green are Sebastian's chromoproteins, that red fluorescent protein and green fluorescent protein. The blue is I took a swab of the roots of one of our like houseplants in our windowsill, and managed to isolate this Pseudomonas species of bacteria. At least I'm assuming it's Pseudomonas, given that it produces this fluorophore pyrovadine that is strongly fluorescent, quenches with iron. Yeah, seems to match. It's a small gram-negative bacteria. Maybe I didn't start most recently enough. Yeah, random looking at pond water, found a little tardigrade. Look at a cute little cutie and a rotifer over there. I wasn't sure what the heck this was, so I took a bunch of photos of it and then eventually figured out it's probably some sort of plant trichome. So unfortunately, the trichomes are usually like dead, too dead to try and like recover this and figure out what plant it's from. But cool nonetheless. It's like little hairy, fluffy sort of cells that are no longer really living cells. Went on a cool hike, saw my first ever hair ice. Dead shrews and sampled some river water there, both to grow out the microbes and also to see what was growing in it. And then when my slide dried out, I found these really cool patterns in the slide. I thought that was a really neat example of just like whatever little bits are dissolved there in the liquid, plus the slow evaporation with all of these random like chunks of dead leaves and animals and so on, leaving behind these incredible patterns. Okay, rapidly zooming forward. Then the other piece that's come up is been getting into tissue culture. Someone got me the Plants in Jars starter kit. And so I feel like a lot of the tissue culture stuff is kind of overemphasize sterility at every step, and also precision in some steps with the hand wavy other parts. Like everyone's micro measuring the exact to make sure it's exactly 1,000 mils for their 1 liter of media, and then they're throwing in about this much in each tub or one chunk of plant, right? So there's a lot of like mismatches. But anyway, so the first thing I tried was like how important is the Instapot or autoclave sterility? So I tried just bringing the media up to I think 96 degrees and then putting it in little tubs. But in pretty much every one, eventually there was like one spore or one tiny bacterium that survived. You could see just this little bubble of contamination that would be a problem later. So good to know that at least probably boiling for a while in a microwave or autoclaving is probably a better bet. Okay, so I've got the Pseudomonas. I noticed under UV, some of these colonies, there's little brown blobs, and in there there'd be this like green fluorescence rather than the bright blue. So I've been trying to figure out if that's a different bacteria growing or just the Pseudomonas aging. I think it might just be the Pseudomonas aging. These are different bacteria, not Pseudomonas. So I've been trying to grow that out, but no results really so far. Here's an example of contamination, in this case from the plant going in rather than from the media itself. So I also, just like I'm going to start cranking up the dial, sterilizing the plants for longer and longer, sterilizing the media more and more thoroughly. But here's an example of like just one tiny little blob of one living thing that still stayed alive in there and was going to grow and cause problems. Um, I've got some beer brewing. It sort of stalled in the ambient temperature down here, which is probably about 16 degrees, but I put a plant germination pad over it to bring the temperature up to about 20, 22 degrees, and it started bubbling away, so hopefully we'll get some life in there shortly. Yeah, so switching to pressure cooker sterilization, I'm testing carrot callus culture. Um, and so for these, I'm varying different amounts of the plant growth hormones, control with no growth hormones. And then for some, I just cut peel the carrot and then cut it into sections and just sterilize it by spraying it with isopropyl alcohol, leaving that for 20, 30 seconds, and then rinsing it off with sterile water. And then for the others, I did 15 minutes agitation in bleach as well on a little homemade stove plate there. The caffeine project, I continue to test new things. This one, I didn't let this stuff dry, so I didn't get such nice patches. I've actually just now, what prompted this video was, it's probably not even loaded yet. Well, by the time we finished, it will. Did a run with some Thai green tea that I just recently bought, and sorry, some Thai iced tea and some pu'er tea. Get a few more samples in. Okay, and then the one thing that I did want to talk about mostly for this video is this idea of biolistics. So I've got aspirations now that I can potentially do tissue culture to try and transform some plants as well. And I can go the agrobacterium route, but I'd prefer to try some of the other non-traditional techniques, partly because there's differences in regulation around using a plant pathogen to do your genetic engineering versus mechanical methods like a gene gun, partly because it seems fun. So the one idea I've had, this is a human hair, and for comparison, this is a 50 micron tungsten wire, right? And that's what the tip looks like if you just cut it with scissors or wire cutters or something. But if you stick that in a blowtorch flame, or a little mini blowtorch, creme brulee torch, you can get it insanely sharp. Right? There's my first attempt, not perfect, but with a little bit of practice finding the right part of the flame, you can just get these incredibly, past my limits of resolving power sharp. This is carrot tissue for comparison. So my idea is to make a bundle of these and see if I can mechanically pierce the same way like the projectiles from a gene gun would, and carry some DNA in with that. Completely speculative, but it sounds like a fun idea, right? Plant microacupuncture or tattooing for transformation, very localized transformation. The other idea is to actually make a gene gun, and I looked at some old DIY gene guns that have been made. It seems like maybe one or two of them got a couple of successful transformations, but no one really did much with them. And they were big and scary and fiddly, right? Because high pressure gas, and you're buying all of these connectors and fittings and valves and solenoid valves. And I realized there's a thing that you can buy on Amazon called an airsoft gun for pretty cheap. I think this one was $37, and it has a CO2 cylinder at 800 psi and a mechanism that very reliably lets out a small pulse of that gas. So I just 3D printed these two little pieces that kind of click on, so I can put it on and cinch, and then fire. And so just to test the spread and the penetration, I put some potassium permanganate on the cling film so that when I shot it into agar media, this is one of the ones that was, one tiny bit of contamination. Why did that go to waste? So let me just blast it with potassium permanganate, and you can see this lovely fall off as in the center of the blast, there's much more penetration than on the sides. So I love that sort of real life data viz aspect of that. Here's just loading a few little droplets of methylene blue as a dye, and then blasting some paper. And I did it so you need some carrier ideally, right, to get better penetration rather than just putting the DNA in liquid. It's not going to get into the cells. And so they usually use tungsten or gold, like really, really small gold microparticles. But that's really expensive and hard to get. So I do have some diatomaceous earth, which looks like it's sort of sharp, spiky, about the right size range. Maybe that'll work. I've also since then found a paper that seemed to use specific type of clay as its sort of microparticle, nanoparticle carrier. So I need something to experiment with once I actually have some DNA. But you can see here I just put a little bit of methylene blue and a little bit of the powdered diatomaceous earth. And then here's a little video of that making sure the safety is on and fired. And then carefully aligning and switching in. I need 3 hands to film. Dot there. Then I'm going to just twist it so that it's locked.
take the safety off, and then put it about an inch away.
Notice that it's pink for a little while and then
There we
fades to
go.
blue. I thought that was interesting. I don't know
Making
whether
sure
it's
the
the
safety is on.
acidification from the carbon dioxide or some reaction with whatever the bleaching agent is, the paper, some sort of temporary redox reaction. I don't know. Anyway, completely aside. The distance you can vary, obviously, to get the speed. And then the nozzle geometry, like you can imagine, like a nice smooth diverging nozzle to use, Coanda effect to spread the air out more, or a converging diverging nozzle to try and get higher speed right above where your carriers are. There's lots of things you can tweak without necessarily worrying about tweaking the pressure or the timing of the release, because those are kind of fixed by the gun. Yeah, so anyway, I think that might be potentially kind of interesting. Okay, there's the potassium permanganate after a while on the agar. Cool color change, and it's now looking mostly brown. Yeah, what else? Okay, so trying more of the needles. What I did was I put along some tape, a few little short sections of the tungsten wire, and then sharpened those in the flame. So you can see there's a couple of sharp ones, and then rolled that up into a bundle, and then twisted them together. So you end up with this like little bundle of spiky needles. And then I tried just needling some onion epidermis. Let me find a better test. So here you can see like I put down 4 dots, but I only needled 2 of them, right? And so then after the needling, you can see those 2 had the needling, and then these 2 didn't. And as a result, the ones that didn't, you just have some absorption where there was, cracks or whatever in the onion skin, but where there's the needling, there's lots more little entry points where you can see the dye's gone in. There's a good illustration of it, just lots of little sections where it's being pushed through. So I'm not sure how deep it'll penetrate. Let's see, I think I did some more experiments later, trying to see like how far below the surface you could get it to go, and I think my wires might actually be too fine. You can see after one little test. But the nice thing is you can sort of just straighten them out and stick them back in the flame, and then you get them all sharpened and shortened pretty easily. So here's another bundle before and after, and the effect on some onion. And you can see like some of them don't really penetrate through below, but some do. Some seem to get past. So anyway, no way of knowing if that's at all a viable method. But for the second bundle, I just wrapped it around a pair of tweezers a few times, and then superglued, twisted, superglued it so that you now have like a bundle, and then cut the other end so you now have a bunch of individual strands. So that was how I got the sort of denser, evenly lengthed bundle, and then just stick the whole thing in the flame. As long as they're vaguely spread out, you can sharpen them all. Okay, some pixie cups. I think that's about. Oh yeah, and so then here's an example of like the media preparation for tissue culture. Got a little stir plate with a computer fan and a couple of magnets and voltage regulator just to change the speed. Hot water, some agar, some MS medias like just plant food, all the key plant foods. And then what I do is add that sort of base media to all of them, and then for some I want them to be callus inducing. So that's you have the cytokinins and the auxins, the 2 different classes of plant hormones. So you put maybe a fair amount of both for callus formation, and versus like rooting, you might want more of the rooting hormone and less, so you can kind of like dial in up and down the concentrations. And I've just started doing this thing where I color code them. So a little bit of red food coloring for rooting, a little bit of green food coloring for shooting, clear for callus inducing. Yeah, one of the carrot tests where I didn't do the bleach sterilization of the so-called explants, the little carrot slices, is already getting some contamination. So it might be that it is necessary to do what everyone does, which is to agitate it in bleach or NADCC for a while. But anyway, we'll see with this latest batch. I also tried, so in addition to the 12 little containers sterilized in the Instapot, I tried pouring some petri dishes after just taking it's like 100 mils of media, sticking them in the microwave so that it boiled for a little bit, as a way to test that as a sterilization method, and then poured that into some sterile petri dishes after adding the appropriate hormones for callus formation. Okay, so to summarize, we got plant tissue culture experiments trying to get callus from carrot as like a model organism for transformation. I currently have no way to get a good plant plasmid to test the transformations, but I'm working on that. If you know, or if you've got some lying around, not that I think anyone's going to watch this video, but if you happen to have access to something like that, please let me know. I am ordering from the Odin. They have a refill for their plant transformation kit, which includes agrobacterium that has a plasmid for ruby. And so I'm ordering that, and my hope is to then mini prep the plasmid out of the agrobacterium, transform it into E. coli, grow it up, mini prep it again to get just the pure plasmid, and then try my transformation experiments with that. But it would be a lot easier to get like a fluorescent reporter gene. So anyway, I'm working on that. Let's see, anything I missed? The penetration test with the diatomaceous earth as a carrier, you see some strong penetration into the media. These little wisps are kind of hard to capture on camera, but without, where's one without? Here, without, it's just surface level. So having those little particles is doing something. I don't know whether these are larger clumps that made it so deep into the media, but again, this is just trying to get intuition rather than actually proceeding to like anything quantifiable just yet. This is all still very early days, probably not going to go anywhere. But I thought I should at least document it, show the pictures. This is onion skin now with the diatomaceous earth plus methylene blue, little 5 microliter droplet on the clingy film, blasted from, I don't know, an inch or two away, just as a test for the transformation. And then this is from the little microneedles, not the gene gun, airsoft gene gun. Anyway, yeah, I hope that's vaguely interesting. It's at least a way for me to use this channel as a kind of backup lab notebook. I also have a physical lab notebook with a bunch of notes and things, but video is a good way to get out the elicit the sort of extra pieces that I forget to write down. So yeah, hopefully that's useful. I will try and mark these videos somehow so that it's not like a proper well-produced video, and intersperse them in amongst the others. But expect in some months potentially some cool plant stuff. Just all takes a while. Oh, and there's the DIY stir bar. Anyone's curious about that. All right, thanks for watching.